Hello friends, Ace Botany next episode. In this episode, we'll be discussing about principles of inheritance and variation, fourth one. What all things we'll be discussing here. For that, I will share the screen so it will be convenient. I think screen came. So now we can proceed to the topic. 7 headings has been given in fourth episode of Principles of Inheritance and Variation. Incomplete dominance, pleiotropy, multiple allism, codominance, lethal gene, polygenic inheritance, and finally, our usual pattern questions, 5 theory, 5 neat model question with the key. So we can start with the incomplete dominance. First, we should know what is the meaning of the term incomplete dominance. In some cases, F1 phenotype did not resemble either of the two parents and is in between two parents. In between. And typical example is NCERT, famous example for theory and runs, a plant called snapdragon, common name dog flower, scientific name Antirrhinum majus, family Plantaginaceae, previous name Scrophulariaceae, after revision Plantaginaceae. Now this is the plant, beautiful diagram, beautiful flowers, etc. Now the cross we can observe, cross we can find here. Red flower crossed with the white flower. Red is capital R, white is small r. F1 is capital R, small r, as usual, here you can find it. But here the result is not red, it is pink. That is how this ratio comes, that is incomplete dominance. Now F1 pink with the capital R, small r heterozygous is selfed. And from each parent we'll be getting 2 types of gametes, so total 4 squares are needed in the checkerboard, and that is what's shown here. And in that checkerboard we can find 1 red, completely dominant, capital R. 1 white, completely recessive, small r. And here you can find 2 pink, capital R, small r. So that is showing incomplete nature, incomplete dominance. And interestingly, phenotypic ratio is 1:2, 2:1. Genotypic ratio is capital R, capital R, small r, capital R, small r. So both the ratios, genotypic and phenotypic, become 1:2, 2:1. It is an exceptional one. Theory question, entrance question also. I think that idea is clear. Now we can proceed to second page. Second page deals with the pleiotropy. Again, what is the meaning of the term pleiotropism? This word pleio came from Greek word meaning is many or more. Here you can find a single gene product may produce more than one effect. Single gene, more than one effect, that's a nutshell. Such a gene is called pleiotrophic gene, and tropos means affecting. So single gene, more than one effect. That is how the word came, pleiotrophy. And a simple diagram shown there you can find gene A, pleiotrophic gene, producing 3 different types of traits. And what is the main reason for pleiotropism? Many scientists believe it is due to the effect of the gene on 2 or more interrelated metabolic pathways that will contribute to the formation of more phenotypes. That is how scientists believe how this pleiotropism affected. An example for pleiotropism, Drosophila. Wing size there is, balancers and eye color. Then gene concerned with the nature of fertility and longevity. Then white eye mutation leads to depigmentation in many other parts of the body in Drosophila. That is regarding Drosophila, 3 items. Now there is a famous disease, specifically humans, name is PKU, phenylketonuria. We have to learn it in disorders. It is an autosomal recessive disorder, chromosome number 12. An enzyme involved is phenylalanine hydroxylase. Details will be studying later. Effect is mental retardation, one effect. Another effect is reduction in hair and skin pigmentation. Single gene, more than one effect. Third is direct NCERT, more predominant for exam. Gene for starch synthesis in pea. It is capital B, small b. Same genes also concerned with the shape of seed, capital B, small b. When we take shape of seed, two shapes are only available. One is round and one is wrinkled. Round will be capital B, capital B, capital B, small b. Wrinkled will be small b, small b. So only two shapes. Now when we take starch grains, its size, three possibilities are there, three. One is large starch grain, completely dominant, capital B, capital B. One is small starch grain, completely recessive, small b, small b. And there is an intermediate size, capital B, small b. So when we take starch grain, that only considered capital B, small b, capital B, small b gives three phenotypic events. So capital B, small b seed show incomplete dominance, intermediate. If seed shape is considered, capital B is dominant and small b is recessive. Seed shape, only two shape, round and wrinkled. Starch grain size, three possibilities are there due to incomplete expression, intermediate expression. That is regarding pleiotropy, and it has got a diagram that I will show you. So it will be easy to follow. This is a famous diagram. Here, long starch grain, capital B, capital B. Here, short starch grain, small b, gametes, capital B, small b. F1 will be capital B, small b. Intermediate starch grain, it is self. And we can find four squares will be in the checkerboard. And there we can find this is capital B, this is small b, this is intermediate. So phenotypic ratio will be 1:2:1. One long, two intermediate, one short. Genotypic ratio will be one long, two intermediate, one short. So we can find genotypic and phenotypic ratio will be same, 1:2:1. It's an exception again. And as such, a monohybrid cross in Pisum sativa in connection with the size of starch grain. In this case, size of starch grain, in this case, we can find genotypic and phenotypic ratio is same, 1:2:1. And connected with the pleiotropism, single gene, more than one effect. An important statement is given connected with NCERT, that is the last page just above the questions. So we'll be remembering it. Now comes next aspect, multiple allelism. What is the meaning of the term multiple allelism? That is, when we look on to Mendelism, gene has got two contrasting forms, that is alleles. But there are cases in which a gene has got more than two alleles, and that event is called multiple allelism. Typical example is ABO blood group system. I think up to that it is clear. Now we can proceed to next page. When we analyze multiple allelism, it acts only in population level. And normally in an individual can have only two alleles for the given gene. So multiple alleles occurs in population, so it can be connected with the population ecology also, evolution. Now comes next term, codominance. And what is the meaning of the term codominance? First, identify codominance is different from incomplete dominance. Codominance means in F1 hybrid that is formed after crossing parents, both alleles express themselves equally. That is the term, equally. Therefore, no mixing of the effect of both alleles. So equal expression, what it further means. That is in F1 progeny that resemble both the parents. And here, alleles have no dominance recessive nature and are able to express themselves independently when they are present together. Separate expression, even though together. It is called codominant alleles. Typical example is different types of red blood cells that determine ABO grouping in humans. I think that idea got what is codominance, how it differs from incomplete dominance, etc. Now, details of blood groups connected with the multiple allelism and codominance. So, all aspect maximum possible is given. Name of the gene is capital I gene. ABO gene is located in chromosome number 9. Capital I gene stands for I designates for a chemical called isoagglutinogen antigen. How many alleles for capital I gene in ABO system? It is 3. They are I, A, I, B, I, O, or small I. I, O, and small I is same. I, A, and I, B are equally dominant. That is why the term codominant. I, O, or small I is recessive. How many main blood groups are there? 4. A, B, AB, O. Number of alleles in an individual, normally it is 2 in diploid condition, 1 in gamete, that is haploid. Universal donor, O group, no antigen, O minus. Universal recipient, AB, AB plus, no antibodies to A or B in AB. How many phenotypes in ABO system? 4. A, B, AB, O. And how many genotypes in ABO system? 6. This we'll be observing in a table in the next page, NCERT. I think this page is clear, data are important. Now we can proceed to next page. This is purely NCERT based data. Once in a while, fill in the blank can be asked. Theory entrance. See, allele from first parent IA, allele from second parent IA. So offspring will be IA, IA. That means blood group will be. This way it is mentioned very easy. Now, how many phenotypes are for multiple alleles? There is a formula for it for multiple alleles to calculate phenotype n plus 1. N is number of alleles, so 3 alleles are there, 3 plus 1 equals 4. How many genotypes for multiple alleles? There is a formula, different manner we can write that formula. Half n into n plus 1, n is number of alleles. So half into 3 plus 1, half into 3, 1 and half, 3 plus 1, 4. So we'll be getting 1 and half into 4, it is 6. I think that idea is clear. Now, what is the biochemical aspect of blood grouping? Different way we can explain. First, we can start with the NCERT. It is pure biochemical aspect, all reactions. What NCERT says, plasma membrane of RBC has got a sugar polymer that will be protruding from its surface. And the kind of sugar is controlled by gene I, capital I. And this capital I gene has got 3 alleles, I, A, I, B, I, small I. Alleles I, A, and I, B produces a slightly different form of sugars, while small I does not produce any sugar. This is given in NCERT page number 61, March 24 edition. Based on nature of sugar, blood group is determined. Simple explanation. Now it's further expansion, more biochemical way when we proceed. Allele IA will produce an enzyme called N-acetyl galactosamyl transferase. Here you can find it. Transferase enzyme, which is able to recognize the fundamental antigen, H antigen in RBC membrane. After recognition, it adds N-acetyl galactosamine to the sugar part of the H antigen to form A antigen. So it is purely biochemical aspect. Same manner, allele IB produce another enzyme, galactosyl transferase. That enzyme will add the chemical galactose. So based on the substance added, the enzyme name came, or reverse. So chemical galactose to the sugar part of the H antigen to form B antigen. Allele IA and IB will produce different forms of sugars. That is why the blood group changes. Allele small I does not produce any sugar, thus antigen also. That is the biochemical aspect. I think it is clear. Now we'll be proceeding to lethal gene. So when we start with the lethal gene, what is the meaning of the term lethal? Lethal means leads to death. These genes or alleles can cause the death of the organism if it carries it. It is initially worked out by a French scientist, L. Cunard, discovered it in 1905. And famous example is coat color in mice, yellow lethals. So lethal genes, different aspects are there. That much details we not need. Still just mentioning only what is lethal gene. Dominant can be there, recessive can be there. It is based on condition. Conditional can be there. And a simple cross is given. 2 yellow mice were crossed. And you can find this is the cross. These are gametes. Simple monohybrid cross, like cross. And we are expecting a ratio 3:1. But what they got is 2:1. Initially, they are not able to get the idea why it is 2:1. Then only they were able to find two Y dominant, yellow dominant comes, that individual die in the early stage itself. That is how the ratio become 2:1. Just to mention what is lethal gene, even though not directly given in the syllabus. Next aspect is polygenic inheritance. Again, what is the meaning of the term polygenic inheritance? See, a phenotypic trait is due to the result of working of more genes. That is multiple genes. Polygenic inheritance has a gradient nature. Gradient nature means tall, less tall, lesser tall, least tall. Same as skin color, dark, very dark, light, very light, intermediate. So this way a gradation can be seen. Typical example is height in humans, skin color in humans. We can easily find the gradation. And the traits are called polygenic traits, generally controlled by 3 or more genes. That is what NCERT page number 69, March 24 given. 3, after 3, I put a star there, we can find can be variable. Normally 3. That is what NCERT says. Role of environment is also influential. Phenotype is due to cumulative effect of each contributing gene. Dominance. Each dominant gene will contribute. So more dominance means it will be more contribute. That is why it is called quantitative inheritance. Typical example is human skin color. And skin color in humans, you know, it is connected with melanin pigments. And connected with the skin color, genes involved are either capital A, capital A, capital B, capital B, capital C, capital C. And reverse small a, small a, small b, small b, small c, small c. Just like a trihybrid cross. And cross between full dominant and full recessive. And when it is crossed, we'll be getting intermediate. That is capital A, small a, capital B, small b, capital C, small c. They are generally called mulattoes or intermediates. I think that is clear. Now we can proceed to next page. Here we can find that intermediates, mulattoes, were crossed because in human selfing is not possible. Two individuals with the same genotype possible. Okay, now capital A, small a, capital B, small b, capital C, small c. 8 different gametic combination possible from one parent, other parent also 8. So 8 squares are needed. That is why 64 squares. When we observe, it will be difficult, but it is comparatively simple. Method I will tell you. All genes, if it is dominant, it is very dark. First one you can find. Dominant. When 5 dominants only, it will be dark. Here you can find any 5 dominants. When it is 4 dominants, here you can find only 4 dominants, that will be any gene, it is fairly dark. And if it is 3 dominants, here you can find it is intermediate. And if it is only 2 intermediate, if it is 2 dominants, not intermediate, it is fairly light. Here you can find only 2 dominants will be there. And if it is having single dominance here, it will be light. And no dominance, very light. So that way you can remember. Very dark, all dominant, dark, 5 dominants, fairly dark, 4 dominants, intermediate, 3 dominants, fairly light, 2 dominants, light, 1 dominant, fairly, very light, no dominance at all. This way you can remember it. To write the checkerboard, no difficulty, you know, just combine. Now very dark will be only 1. Dark will be 6. Fairly dark will be 15. Intermediates will be maximum with 20. Fairly light will be 15. Light will be 6. Very light will be 1. So intermediates maximum and these 2 extremes will be minimum. 1 each, so total 2 out of 64. So ratio become 1:6:15:20:15:6:1. Total 64. And when we plot this, here you can find it will be having a bell-shaped curve with the center one maximum representation. It is intermediate or mulattoes. So maximum will be intermediate or mulattoes, 20 out of 64. Minimum will be 2, very light, 1/64, very dark, 1/64. To find number of phenotypes in polygenic inheritance, there is a formula 2n plus 1, n equals number of polygenes. 3 polygenes, so 2 into 3 plus 1, 7. To find number of genotypes, 3 raised to n. So 3 raised to n, that is n for number of pair of polygenes. Here written clearly. So 3 into 3 into 3, it is 27. So total number of phenotypes and genotypes in human skin pigmentation will be 7 phenotype and 27 genotype, total will be 34. I think that idea is clear. Since polygenes given in NCERT book, this much details not mentioned, but still it is good if you know all this. Now, as usual, 5 theory question, 5 NEET model question. I think with the key, idea is clear. So what all things we learn? Almost 7 topics, of which last topic is question theory entrance. Now, starting with incomplete dominance, pleiotrophy, multiple allelism, codominance, lethal gene, polygenic inheritance, and question. And this is an area where questions usually comes for theory as well as NEET exam. Very, very predominant area for exam point of view. Comparatively easy, no difficulty, I think. Lethal gene, just mentioned to know such a factor is there. I think idea clear. If you like this episode, subscribe all, like it, share it, comment on it. We can meet later with another episode. Thank you.